Good afternoon, everyone. Thank you for joining us. And before I start, I want to thank everyone here and online, everywhere I'm thanking on behalf of Balu and Dr. Kirsch for helping us to put together our renewal of the FES Center, which was successfully submitted last week. And hopefully a few months down the road, we will announce it was successfully awarded, and then we'll clap again. So thank you for that. And this is our final seminar until the fall, but stay tuned for more detail for summer neurotech talks, which will be coming soon. And for today, I am announcing a special guest, Professor Jeff Schall, who is here from York University in Toronto. We have known Dr. Schall for many, many years, and he actually visited Cleveland. He visited our lab about 25 years back when my mentor, John Lee, was around. And he has been long-term colleague of many of us, and at that time he was at Vanderbilt. So Dr. Schall is a director of neurophysiology and visual neurophysiology center at York University. He joined York Department of Biology in 2021, and prior to that, he was at Vanderbilt, where he was a professor in Department of Psychology and a Bronson Ingham Professor of Neuroscience. He was also the founding director of Vanderbilt Center for Integrative Cognitive Neuroscience since 2000, and from 1998 to 2005, he also directed Vanderbilt Vision Research Center, during which he oversaw staff, services, and trainees to facilitate vision and eye research across departments and schools of Vanderbilt. He's internationally recognized as a scholar in the field of visual neurophysiology, earned his PhD in anatomy at University of Utah School of Medicine, and then he had postdoctoral training at MIT. His research was supported by grant from the National Institute of Health, National Science Foundation, Air Force Office of Scientific Research, and now the National Sciences and Engineering Research Council in Canada. His scholarly accomplishments have been well recognized with numerous awards, including our Alfred P. Sloan Foundation and James H. McDonald Foundation, McKnight Endowment Fund in Neuroscience, as well as he received Troland Research Award from United States National Academy of Science. He is a fellow of Association for Psychological Science and American Association for Advancement in Science. He was the president of Vision Science Society in 2009. And he is currently a member of Center for Vision Research and Vision Science Application VISTA program, which is funded by Canada Force Research Excellence Fund. Without any further delay, Professor Schall.
Good afternoon. It's been a great day. I've enjoyed being here, meeting the faculty and the students and seeing the amazing facilities. So I hope this talk is useful and interesting to you. I'm going to be talking about a set of work accomplished with a large number of very talented trainees and faculty colleagues. That's an electrocardiograph when it was invented in 1902. As you know, the ECG is very useful, very informative. The PQRS wave. We were talking about that with some of the students today. It's useful in the clinic just because we know how it arises from the actions of the heart. So that if the P or the Q or the R or the ST wave are different in some way, cardiologists can identify what's wrong with the heart. I understand it's the most useful or common bedside diagnostic tool in use in the clinic today. And you can buy a little ECG monitor on Amazon and check your ECG. Very useful electrical signal. This is the first EEG, first electroencephalograph in 1924 that Hans Berger used to record EEG in humans. It's useful, tells us if you're awake or asleep, tells you if you're having an epileptic seizure. It's not irrelevant. We don't, we can use it to some extent. And some EEG measures from the nervous system are really useful. But the biophysics of the origin of EEG is understood in some outline, that when pyramidal cells in a part of the cortex discharge, it sets up a polarity with positive and negative charges separated, which creates a little battery, which generates current, which can make voltage on the top of the head. So that basic physical principle is well understood. In other parts of the nervous system, the electroretinogram has a high degree of clinical relevance and utility, as does auditory brainstem potentials. Again, just because we know where they come from. The different waves come from different cell nuclei, and that makes the identification of disorder or of targeted treatment more useful. But EEG is still a mystery. A colleague wrote, it's such a mystery you can write a review for Transit Neuroscience that says, where does EEG come from? What does it mean? We know shockingly little to answer this question. And yet it's used on the bedside for epilepsy patients. And you can buy one on Amazon and improve your life, sleep better, be happier, make more money. I don't think it'll make you better looking, though. So we tackled this. Jeff Woodman was a postdoc in my lab at the time. He's now on the faculty at Vanderbilt. And we said, well, do monkeys have EEG? Sure they do. But what do we know about it? Other than a few studies in the early 1980s, late 1970s that showed that monkeys have a P300, an event-related potential called a P3 that's like beep, 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 boop. On the boop, you had a P3. So we arranged electrodes on the surface of the cranium, not the head, not the scalp, because unlike us, macaque monkeys have a lot of muscle on the top of their head, which would corrupt the signal. And so we put the electrodes on the surface of the head, and we used international standard known as the 1020 system that's labeled there to position the electrodes, just to have some kind of uniformity across the different animals. One of the first event-related potential components we investigated was the N2PC. Second negative wave, posterior, contralateral to where you're paying attention. It had been discovered by Steve Luck when he was a graduate student, and Jeff Woodman was a student of his, so we knew what the N2PC should look like. We had monkeys do a visual search task. In blue, he's looking for what is he looking for? He's looking for an L among T's. And so you can see in the blue array, the L is on the right side. So the left hemisphere is more polarized because he's paying attention on the right hemifield. In red, the L is on the left side of the visual display. So the right hemisphere is more polarized because he's paying attention to the left. And the EEG patterns that you see labeled there show that difference. That difference occurs when the monkey's allocating attention. That's the N2PC. So monkeys have an N2PC. Based on the distribution of voltages on the head, it's possible to do inverse solutions to calculate where in the brain the currents could be to generate that pattern of voltages. And one solution is shown there on the upper right. There's a hot spot over the back of the brain, contralateral to where the target is. And the hotter the color, the more presumed current was flowing in that area. And it's a sensible part of the brain. It's V1 and V2 and V3 and V4. I mean, those are the right parts of the brain for it to take place. The problem is with these inverse solutions, and those of you who've had the math understand how there's not a unique solution. Multiple current sources can generate the same voltage distribution. And so you have to make certain assumptions, and you can argue over those assumptions. And for some people, like Steve Luck, because this is unsolvable, he's not interested in it. We'll just use the EEG as is and punt on the question of where in the brain does it come from, because it's impossible to answer in humans because you can't get the information you need. With monkeys, we can get the information you need. Yeah, the N2, we talked about this earlier today with some people. So the relevance of the N2PC as a biomarker is it's abnormally large or abnormally smaller in different patient groups, such as those with attention disorders and cognitive impairments leading to dementia. So it's different in patient groups. How it gets to be different seems to be an important question on understanding what it is telling us as a biomarker and possibly informing us of what therapies to do or where to do them. But at the moment, we don't know where it comes from. In 50 minutes, you will. We looked at another ERP component called the error-related negativity. It was discovered simultaneously in the US and in Germany in the early 1990s. And it's been known as the blunder blip because it happens when you make a mistake and feel oops. And so we elicited it in a task, I was mentioning this to a few people today, called a stop signal task. In our version of it, the monkey fixates a square in the center, spot jumps, and he looks at it. Or it could jump the other way and he looks at it. And he does that many times, and we get a distribution of reaction times. But on a few trials, ready, set, go, and then the central spot comes back on that says stop. If the monkey doesn't make the movement he was going to make, he gets his juice, or she gets her juice. If they make the saccade, no juice. That's the error. And so that what's plotted at the top is the EEG on the top of the head, same place in monkeys as in humans. The solid line plots the activity on trials that were correct, and the dotted line or dashed line plots the activity or the polarization, I should say, on trials when the monkey made an error. That small light gray area here is illustrating the ERN. It's just a little voltage difference. It's not very big in U either. After that, there's an excessive positivity that's also characteristic of this event-related potential component in people too. We can do the same inverse solutions, and we get hot spots in medial frontal cortex, even going down into the medial wall a bit. But remember, these are not unique solutions. If we change a parameter or we change an assumption, it's going to become somewhat different. It's not going to go to the back of the brain. It's not that different. What does this look like in humans? Here's a picture. So Jeff Woodman's lab did the exact same experiment, the very same tasks in human volunteers, measured EEG in the same way and applied exactly the same pipeline to it. And these are showing the inverse solutions of the distributions of currents making the ERN in people. Doing the saccade task on the right, and then doing a version of the task where they're responding with their hands on the right. And so this error system isn't particular to effector. You can have oops with any part of your body, right? And this biomarker is relevant for impairments of self-monitoring and executive control. Every psychopathology, for example, and many neurological disorders are characterized by these issues. So, what can we do in the monkey that you can't do in a person? And this is what we can do. We can put a microelectrode in the brain and an array of microelectrode contacts and sample signals across all the layers of the cortex. And so on the upper left is a MR showing where we were targeting. On the right in B is showing the exact location of a couple of penetrations of these microelectrode arrays in what's known as the lunate gyrus in macaque monkeys, which is where an area, the V4, the fourth extrastriate visual area, lives. On the lower left, this is a plot, well, a cartoon of the electrode, but then a plot of the receptive fields of the neurons sampled at different depths. And the receptive fields all are on top of each other, which confirms that we're going in perpendicular to the layers. If we were going in at some angle, the receptive fields would move across the visual field. B and C plot a quantity known as current source density. So on each contact, the voltage is fluctuating. We can align all the voltages on the presentation of the array, and you know Ohm's law, right? E equals IR. So we have a sense of what the resistance or the impedance of brain tissue is. We have voltages, we can solve for current, and that's the current density. The red parts are the current sinks where the depolarizations are happening, and then the blue parts are the current sources. B is showing one session, C is showing the average of many sessions, and they're not that different. Because the current source density in an area like V4, when you're flashing lights, is a very characteristic, has a very canonical sort of appearance. The lower right just shows where the receptive fields were sampled in 2 different monkeys. All right? So we can put a linear electrode array across the layers of the cortex, and with that information, obtain a density of current in association with some event. In this case, it's presentation of the array. Now you can see that the initial period, the first depolarization is happening early in this middle layer. That's layer 4. That's the input layer of this visual area, this part of the cerebral cortex. So the visual inputs come into layer 4, and then they're sent to the upper and the lower layers. So this long-lasting current density in the upper layers, layer 2 and upper layer 3, well, that's the brain processing the image in some sense. What's being processed in computational terms, I'll tell you about in a minute. So we're going to tell a story about V4. We're also going to tell a story about supplementary eye field, an area in the medial frontal cortex. This again is showing CT and MR co-registered to demonstrate that we're going in perpendicular to the layers. You can see the electrode and the voltages sampled, the density of gamma band activity, the density of spikes isolated on each contact, and then a current density aligned on the visual event of the target jumping. Now you'll notice the current density on the right looks different from the current density on the left. SEF and V4 look different. And we weren't sure when we started this whether we would get to see anything like what V4 or V1 shows in current density. Fortunately, it did enough for us to proceed with the study as I'm going to explain it to you. So I'm going to tell you a story about supplementary eye field as well. In both areas, we're sampling across all layers. In both areas, we can calculate current density. And from that, we can determine the dipole making the EEG. So let's do V4. Here is, this is the task the monkey's doing. He's fixating, an array of objects comes up. One is red, the others are green. He's supposed to look at the oddball, look at the red one. All right? And that's it. It's very simple. It doesn't take a lot of time for them to learn this. And we can contrast 2 trial types. In one trial type, the target, the thing he's going to look at, is in the cell's receptive field. In the alternative condition, the target's somewhere else, and the thing in the receptive field is the distractor to ignore. All right? And so the top current density is showing the pattern when the target's in the receptive field, when the distractor's in the receptive field, and their difference. So we see when we subtract one from the other, that early sink in layer 4 is gone because it's equivalent strength, whether it's a target or a distractor in the receptive field. And only later does the current density differ. Only later can you allocate attention to the target. It takes time to sort out what's where in the array. The panels on the bottom, oh, I forgot I animated this. So there's CSD for target and distractor, their difference. So Jake Westerberg, who did this experiment, used information theory approaches just to demonstrate that there's a statistical association between the spikes in the brain, in V4, and the voltages on the head. Trial by trial, neurons fire different numbers of spikes. Voltages fluctuate a little bit. The variation of spiking activity in V4 predicts or is associated with the current density observed in layer 4. All right? And its association with the N2PC. The association with the N2PC is stronger for the neurons in the upper layers, above layer 4. It's weakest in layer 4, and it's stronger again in the infragranular layers 5 and 6. That's a theme, and we're going to see other examples of it, that the association of an area with the EEG is laminar specific. Certain cells have more to do with the EEG than other cells. That seems to me to be a relevant fact of the matter for thinking about this as a biomarker. Now, we can do the inverse model. As I said, it's an indefinite solution because multiple solutions are possible. So let's do the alternative. So if I say given voltage, where are the currents, it has an indefinite solution. If I say given the currents, where's the voltage? If I do the forward model, that has a definite solution, one and only one answer. And so with Jorge Riera and Beatriz Herrera at Florida International University, they had the capacity to do these kinds of analyses. And so to do the forward model, you need a model of the head and its geometry and conductances. And so this is showing how that model was developed, a boundary element model of each surface between the electrocontacts and the head, the surface of the head. And these boundary element models can be as precise as we need them to be. They can account for the size and shape of craniotomies, for example. We were talking about this earlier today, about what influence that would have. And so here's the same display. We've got the target in the receptive field, the distractor in the receptive field. And what's plotted on the bottom now is the dipole strength under the two stimulus conditions derived from the current density. That was recorded from that location on the lunate gyrus. Okay, so you see the black dot there. And here's what you get. If I put that current at that location in the monkey's brain and say where are the voltages resulting from current in just that one location, the answer is shown here for target in receptive field, distractor in receptive field, and their difference. And I hope you appreciate that this is looking like the N2PC. It's the second negative posterior contralateral. But that's from one site in the brain, one and only one site. And of course, the brain doesn't work like that. The brain's a distributed process. So what does it look like for another part of the prelunate gyrus, or the lunate gyrus? It's about the same. And yet another location that's a little bit further away. I mean, it's a little bit different, but it's, globally the same. Now, V4, the area known as V4, is on the surface of the gyrus, and it also goes down the medial, down the sulcus on the front. We can't record across the layers down there, but we can make the conjecture that the current density is going to be the same in the sulcus as it is up on top. And so we put the current down there, and we get this appearance. Again, it's a contralateral emphasis of current opposite the location of the target. All of these were active together, so we can add them all up, and that's going to be the N2PC, right? Well, there's other areas that are doing the same thing as V4. So this is showing data published by other labs. On the upper left, it's the activity recorded in an area called LIP, the lateral interparietal area in posterior parietal cortex. On the right is the, in a raster display, and the spike density function, the firing rate when the target's in the receptive field. On the right is when it's a distractor. You have to superimpose them in your mind's eye, but you can see that you get the same motif of common activation followed by a separation, greater activity for the target, less for the distractor. The lower plot is the same kind of thing from area 7A, another region, it's on the convexity of the parietal gyrus there. And so we can conjecture that the current density is the same in those areas. We don't know, but if you'll allow me that assumption, I can put our dipole in these areas and ask what's the distribution of voltages you would get, and there's LIP, and down here is 7A. So again, there's a contralateral nature to it. But there's another area that also is active in visual search, and that's the frontal eye field. Up in the front of the brain, it's on the rostral bank of the arcuate sulcus, is what it's called in monkeys. It's about the same location in us, but we call it the precentral sulcus. And frontal eye field is connected with and interacts with V4 and 7A and LIP. It's a distributed network. If you stimulate frontal eye field, Tiernan Moore showed that you can influence attention and the state of activity in V4. When we record in frontal eye field and at the same time as the EEG with the N2PC, here's a summary plot. In blue is the firing rate. The most saturated is target and receptive field. The less saturated is distractor. So we again see it's common activity and then it separates for the spikes. In green are the local field potentials recorded in frontal eye field. And we see that same motif of a separation. And at the bottom in red is the N2PC again. In this case, the monkey's doing this T among L or L among T search. So it takes more time for him to locate the item because there's a similarity. We also see that the spikes modulate in frontal eye field before the N2PC arises, raising the possibility that the N2PC is influenced, maybe not caused by, but certainly frontal eye field is in the game to be influencing the network to locate the target among distractors, which is where you're allocating attention, which is what N2PC is indexing. Does frontal eye field contribute biophysically? Oops, sorry, let me make this point. And there's a statistical association again, just like that information theory. This is just straight correlation. So on a given trial, you get so many spikes, and it varies a fair amount. On a given trial, you get a certain polarization, and it varies some amount. The trial-by-trial correlation of FEF local field potentials is associated with the N2PC. The spikes weren't in this case, but the local field potentials in frontal eye field were modestly predictive of what the degree of polarization of the N2PC would be. So there's an association, right, anatomically, functionally, of frontal eye field with V4 or whatever areas are generating the N2PC. We can do this biophysical trick again. So we're going to pretend that we have our dipole in frontal eye field, because I know where frontal eye field is. And I said, Beatrice, put the dipole there. And she did. And that's the current pattern that you get. It's pretty weak. It's lateralized, but it doesn't look like the N2PC at all. So now we got frontal eye field, we got LIP, we got 7A, we got V4, we got different parts of V4. Let's just put them all together. And that's what Jake and Beatrice did. So this is a histogram of R-squared values. We have the EEG at each electrode on each trial observed, and then we can do the predicted by the forward model, including a dipole in each of these areas and every possible combination. So the best prediction of the N2PC came by combining the V4 out on the gyrus, the V4 down in the sulcus, and LIP. Frontal eye field didn't contribute much at all biophysically to the N2PC. All right? And so, what we've demonstrated, it's the most representative model. So, an interim summary. Area V4 contributes directly to the N2PC. Now, human studies have come to a parallel conclusion. Under these testing conditions, the spatial pattern of EEG was explained by summation of sources in V4 and LIP. So the sources of an event-related potential don't have to be adjacent. They can be kind of millimeters apart from each other. Areas like frontal eye field can contribute computationally, neurocomputationally, but not necessarily biophysically. So the ERP is not showing you everything that's going on. And now with information about sources from fMRI and reasonable assumptions about cortical dipoles, you can arrive at similar conclusions for humans. We don't know what the dipoles look like because it's not been possible to record across all layers until relatively recently, but not as systematically as you can do in a monkey. You with me? Yeah. All right. So I told you where the N2PC comes from. What does it mean? What is it telling us? How would you interpret it? Now, in the brainstem saccade generator, as we learned from David Robinson, we use words like integrator. We describe part of the circuit as an integrator because we understand that that's the thing that that network is doing, right? What is V4 and the rest of this network doing? What is the N2PC telling us about? Well, those are questions that psychologists answer, right? And so what you're looking at is a model framework developed by a cognitive psychologist in Melbourne, Australia, Philip Smith, in which it's a model of attention. And he's got a where pathway and a what pathway. On the what pathway, it's going to tell you, is it a T, is it L, is it red, is it green? The where pathway is sorting out where things are and where am I going to allocate attention. None of this is on the test. Don't worry about that. But this what and where pathway, of course, are just the dorsal and ventral streams in some sense, and they both converge in frontal eye field. So, with Gordon Logan and Tom Palmieri and a talented postdoc, now faculty member at SUNY Albany, or now it's called University of Albany, Greg Cox, we did a model of how this salience representation arises. All right, so I got to tell you this story to answer the N2PC story. And so again, there's a complexity here, but it's not that complex. This will simplify it for you. So it's a differential equation. The rate of change of the M units there is dictated by these quantities. There's a decay term, so it doesn't integrate to infinity. And then it's the activity feeding it. There's just that one red arrow or black arrow feeding into it. It's that activity minus a gate. So there's a gate that prevents integrating noise. All right? And then there's lateral inhibition, and then there's stochastic noise. All right? So we can get a reaction time out of it. There's this component which is identified. It is the identification component. And so those equations have a recurrent gating. There's a certain latency. There's lateral inhibition between them. What these units are doing is specifying what the target is. Like, is it the red thing? Is it the T? Is it the green L? Right? So there's a lot of intelligence in here that also needs to be explained in subsequent models. But there's an identification component and a localization component, which is just where the lights are. And then this is the salience evidence. This is the equation we think specifies what the FEF visually selection neurons are doing. And so again, it's a differential equation. We're just driven by the inputs. There's feed-forward inhibition, there's identification feed-forward inhibition, and then lateral inhibition as well. So we have the tools, the computational tools, there's a lot of parameters here. So how do you navigate that space? And that's where Tom Palmieri has the expertise to fit the model, to minimize the Bayesian information criteria, to identify which components or which parameters are driving the fit. Okay, now what are we fitting? We're not fitting behavior with this. This is a cognitive model, but what we're going to fit are individual neurons, the spike densities of individual neurons in frontal eye field. And I wouldn't be telling you about it if it didn't work. So at the top is a dendrogram that organizes into three groups. the sets of parameters, the parameters are listed on the left, recurrence, identification delay, so on and so forth. Those are all the parameters. And where there's a kind of a filled-in space, that neuron needed that parameter to fit its activity. And they group into these interesting groups. So here are what the neurons that are driven by the identification lateral inhibition. As it happens, the lateral inhibition between the identification units was an important thing. It was necessary to fit the activity of these frontal eye field neurons. So the thin line is the observed data, the real jiggly line. The smoother line running through it is the simulated data. You can see there's a very high quality fit for set size 2, set size 4, set size 8 of T among L's, and then the other two are, red among green, let's say, and red among not so red. All right? So we're fitting individual neurons. There's a set of neurons for whom lateral inhibition between the identification units was crucial. There's other elements in here too. I'm not going to go into all the details. Here are the neurons for whom recurrence was necessary. You see it's a good quality fit, but you see the dynamics, they look different, right? The dynamics of these two pools of neurons are not equivalent. They're not interchangeable. And then neurons that don't need either of those but have other components are fit as well, but they're not, their dynamics are different too. So as a neurophysiologist, when we record from neurons, we recognize members of families. We see neurons that do this and neurons that do that. They tend to get described under these umbrella terms because we don't have any reason to distinguish them yet. This is one of the first instances of a computational model predicting differences among dynamics of neurons. Now, in subsequent work, we want to know, are these, are some in layer 3 and others are in layer 5? Are some interneurons and some are pyramidal neurons? I mean, there's a lot of open questions suggested by this. But I hope you'll allow that this model does an explanation of what the FEF neurons are doing. So while we were recording these neurons, we had EEG leads still on. And so here's the next new bit Greg and another talented Korean postdoc, Giwon Bog, did. So we want to predict the N2PC. And we've got the frontal eye field activity recorded at the same time, described in terms of the parameters of this model. And so we happen to have 59 neurons satisfying a variety of criteria, including being sampled with the EEG. And then we have the EEG over both hemispheres. And so what this complex figure is showing is simply that we've got the list of parameters of each neuron, and we can predict how well it will predict through regression the ERP for both hemispheres. Okay? There's a complexity here that we don't want to get hung up in. The punchline is, based on the activity of frontal eye field neurons, we can predict the N2PC. And indeed we can. So this is showing the N2PC observed and predicted for set size 2, 4, and 8 in the TNL search. The N2PC is smaller and later when there's 7 distractors as opposed to 3 as opposed to 1, because it takes longer to find the target under those conditions. That's known from the human literature. We saw it in the monkey data, and we're modeling it here. So we predict the latency and the amplitude of the N2PC from the nature of the activity of neurons in frontal eye field. Here's something we didn't expect. So this regression, it's a linear regression. So you get all these different components. They're going to add up to make the N2PC. And so for set size 2, 4, 8, the purple line is showing the identification parameters, how it varies through time. L is the localization parameter. S is the salience parameter. You see how the I is going positive and the S is going negative? Every model that fit the neurons required those two parameters to have opposite signs. So the I minus S plus was pretty good too. I plus S minus was best of all. But I minus S minus or I plus S plus didn't fit very well. It fit okay, but didn't fit as well. What does that mean? How could these parameters have different signs? Well, here's a speculative conjecture. Whoops, what did I do? Yeah, so remember the N2PC. Remember I told you it came from V4 and LIP? These are MRs through monkey brains showing the physical location of LIP and V4. LIP is painted in kind of a blue. V4 is painted in a kind of orange or yellow. Geometrically, they're pointed in opposite directions. V4 kind of goes this way and LIP kind of goes that way. So could it be that the identification and the salience parameters have opposite signs just because they're driven by activity in different cortical areas? We don't know that yet, but it suggests specific things we can look for to test. Okay, that's a biophysical and computational explanation of the N2PC. Any questions? Okay, let's do the ERN. I'll go quicker. So the supplementary eye field doesn't have layer 4. It's like primary motor cortex and premotor cortex. There is no layer 4. And so one of the animating issues for this work is what's the microcircuit? We've worked out a canonical cortical microcircuit from primary sensory areas, and some people have just said, well, that's the cerebral cortex. Just propagate that everywhere. Won't work here. So in histological sections stained for acetylcholinesterase or myelin or Nissl, on top is primary visual cortex, and you can see the strong layer 4. And in SCF, you see it's just not as laminated. It just looks different. And nevertheless, we could work out the depths of the linear electrode arrays and calculate current density as we did before. It looks somewhat different. There's a story to say there. I told you we're going to look for the ERN. We recorded ERN at the same time as the recordings with the linear electrode arrays. The ERN, I told you, was discovered in 1993. The first attempt to locate it, Stan DeHaan did, using these inverse solutions, and identified it with anterior cingulate cortex. And a bunch of other follow-up work in this domain emphasized anterior cingulate cortex, principally because anterior cingulate cortex was mysterious, and you get more points for working on mysterious areas than non-mysterious areas. So students, write that down. Work on mysteries. If you work on stuff everyone understands, no one believes you. Okay, so we had this stop signal task, as I explained to you. And we had worked out how the motor circuits do it. Then we went back to supplementary eye field, this area on the top of the head, that's the eye part of supplementary motor area. And here's what we found. In black, this is the activity of one neuron. Black are trials when he makes his saccade correctly, there was no stop signal, he got his juice, to the right or to the left. The red line, the red interrupted line, is showing the activity on trials when he made the same saccade, but it was an error. And you see the strong modulation. That's the error-related signal I want to talk about. And so it was found in supplementary eye field. But wait a minute, didn't I just tell you anterior cingulate is supposed to be where the ERN comes from? Yeah, that's where it comes from. So we advanced the electrode down into anterior cingulate cortex. And we see the same kind of error responses there too. So there may be two sources. We also had the wherewithal at this time to do another manipulation where the monkey does it right and he gets his juice. So the bottom plots here are now plotted on the time from reinforcement. Right, so he knows he's going to get his juice. Black lines plot the trials when he got his juice. We deliver the reinforcement. The interrupted lines are showing what happens when he did it right. He was supposed to get his juice. He just didn't give it to him. And you see the neuromodulation. It's like, oops. It's not oops, I did something wrong. It's like, oops, I didn't get what I was supposed to get. Or what people refer to as a reward prediction error. So those signals are in both places. They're also in you. I think they're in you. When you have epilepsy, you can go to Cedars-Sinai and we'll see. Because our colleagues, Uli Rudeshauser and his group, are recording in humans in these medial frontal areas in similar tasks where they make errors. On the left, well, we wrote this review together to try and see if the human and the monkey are similar. And if they're different, in what ways. So one way they're different is in monkeys, I can get 1,000 or more trials a day for many, many days. For humans, you don't get that many trials and they're out of the hospital in a week. So you have a very limited sampling time. On the left is a monkey error neuron. On the right is a human error neuron. How do you quantify if they're equivalent signals? Well, we did this area under the ROC, constructed it for correct versus error trials. And the area under the ROC for humans and monkeys is equivalent. So even though you're sampling fewer trials, it looks like it's capturing the same signal. When we put linear electrode arrays in supplementary eye field, we can begin to ask, where are the error neurons? Are they in one layer, two layer, all layers? Where are they? And so what's plotted on the bottom is what we call a time depth plot. So time is synchronized on the time of the action, the saccade. The symbol plots the moment when the activity became significantly different. A band continues until the activity returned to normal or quit being different. Triangles are wider spikes, slower repolarizing spikes, and the star symbols are the fast spikes. Maybe they're interneurons. You can see that there's error-related activity all the way through in each layer. Seems to be kind of dense in layer 3 and upper layer 5, and it's earliest in lower 3 and upper layer 5. Don't know what it means, but when Hublin Wiesel recorded from visual cortex and did plots like this, no one knew what it meant then either. The state of knowledge of the frontal lobe, the laminar sort of functional architecture of the frontal lobe, is where visual cortex was in 1958. So we've got a long way to go. Now, we can, as I said, we're recording across layers. We can, at the same time as the error-related negativity, we can ask, is there a statistical association of the spiking activity in supplementary eye field with the fluctuations of the ERN? Answer is yes, there is, but only for upper layer cells, layer 2, 3, not for layer 5, 6. Don't know why that's true, but that turned out to be the case. So there's a statistical association, just like V4 to N2PC. But we can get current source density from supplementary eye field. They're different in error versus correct trials. So there's a basis for a dipole here that could predict the ERN. So Beatrice did it. On the left is observed, on the right is modeled or predicted dipole from current density in supplementary eye field alone. There's a little ERN there. I'm not going to leave this up long enough for you to scrutinize it because there are some embarrassing differences here. So SEF doesn't really explain all of the ERN, all right? But they're not equivalent. Where else could it be? Well, anterior cingulate cortex. We're back to that again. But as I was explaining to some folks today, this is a portrayal of the dipoles that would exist in supplementary eye field on the top, and then on the medial wall, and then dorsal bank of the cingulate in monkeys, and then the ventral bank of the cingulate in monkeys. And so they're pointing in different directions, which are going to cancel and summate. There's a complex biophysical sort of problem here. It's not insolvable, though, certainly. It's just more math. Now, in humans, this becomes critical because some humans have one cingulate sulcus, others have two. And you might have one in one hemisphere and two in your other hemisphere. It's just really variable. So if there's two, you've got even more folding to account for in the geometry of the dipoles predicting the error-related signal. Are there error neurons in all of these areas? So this is other new data. We're plotting activity from supplementary eye field, from the dorsal bank of the cingulate sulcus, and from the ventral bank of the cingulate sulcus. You've never seen this before. Nobody's ever done this before. On the left are neurons that are facilitated, fire more on errors. On the right are neurons that are suppressed or fire less on errors. And the color bands are showing where the hot activity is, kind of a raster of all the neurons. They're ordered by their latency. We see that there's lots of errors. There are error cells in all three areas. Maybe there's fewer in ventral cingulate than dorsal cingulate than supplementary eye field. Okay. We also notice that the latency in supplementary eye field is shorter. The error signal arises sooner in supplementary eye field than in the cingulate. These cells carry other signals. I'm not going to go through this. I'm just showing you that there's lots of other aspects of this neural activity we can be thinking about to do the same sort of computational model that I told you about for the N2PC. And so, with simultaneous recordings of EEG and laminar spiking with local field potentials, we can show how V4 contributes to the N2PC, how SEF with other areas contributes to the error-related negativity. And so the cortical sources of cognitive ERPs can be located in macaque monkeys. Specific neural spiking signals in certain layers are associated with variation of ERP polarization. And that laminar information may be useful, I hope it's useful, at some point for the clinicians in designing treatments or understanding the signals as biomarkers. And then forward modeling can be done on the basis of getting the right current density information at this mesoscopic level and forming dipoles and doing the forward models that will have definite solutions. Cortical areas can contribute computationally, but not biophysically. So remember, frontal eye field has a lot to do with finding targets during visual search. The same thing N2PC's indexing, but frontal eye field doesn't generate any biophysical signal there. And so the N2PC or any event-related potential component may not reveal properly the underlying timing of constituent operations. And then you got to distinguish agranular from granular cortex. So general conclusions. There's the good news. Cortical sources of cognitive ERP can be located, even if they're in non-adjacent areas. That's the good news. The bad news is, for multiple reasons, there are multiple reasons for heterogeneity of an event-related potential across the individuals and diagnosis. You know, so if you see that let's say the ERN is smaller in this person, is it smaller because their folding pattern's different? Is it smaller because their pyramidal cells are weaker? Is it smaller because they have fewer pyramidal cells? It's undecided. But the list is not uncountably large. So there's very specific things to look at to understand why an ERP component may be larger or smaller than normal. And then the promising news, I think EEG can become as informative and useful as ECG. So that's what I had for you today. Thank you for your attention. Yes, sir. I see we got.
Oh, fantastic, Jeff. Your last comment about the EEG being a useful thing, this is a response to a stimulus, a very discrete stimulus. Yeah. So how do you generalize that? I mean, people use EEG for a lot of different things.
Right.
And maybe more complicated. Is there a pathway to generalize what you've done?
Yeah, we've talked about that. I mean, experimentally, we've got to synchronize things or we can't even get off the ground. But let's say we figure out something like N2PC, and it's this set of areas. Then we can, on the one hand, record from those areas while the monkey's doing nothing. or while the monkey's in whatever state your patient's in, I mean, that I can do with the monkey, and watch how this thing fluctuates. Because now we know where to look for the cortical sources of that EEG thing. Will they predict it all the time? Maybe they don't predict it under, let's say, resting state conditions as they do under the stimulus evoked condition. That's one approach to it that we've thought of. But it's a very important question, and again, tractable.
So, there's a lot of BMI work, and having a reliable prediction of, whoops, I didn't mean to do that, would really help a lot of different things. And given what you have covered and know this is something that would need to be on a single trial,
Yeah.
the ongoing asynchronous, oops, I didn't mean to do that, situation. Knowing everything you know, what would be your ideal type of electrode and target and type of signal that you think is going to be the most likely candidate to get a, oops, I didn't mean to do that, in a real-time BMI control situation?
Yeah, I would, we talked about this today, I'd put a Utah array in the top of the medial frontal cortex.
Okay.
So for eye movements, the supplementary eye field in humans is on the medial wall. And you don't put electrodes on cell side at all like that, yeah. So you'd have to be up on top, but that's okay for the arms and limbs and mouth. It may be out on the surface. That's the first place I'd start, and then decode it with an appreciation that you're seeing not motor command signals, but kind of monitoring signals.
And what do you think is can you speculate on the type of reliability that might be possible?
I don't know. In our situation, because we have so few contacts, we have to sort of add up.
Yeah.
I mean, I think if you get to 20 cells, you're going to be pretty good, like 90% or so. In each of the things where we've looked at how does a signal improve with the number of units contributing to it, or the number of trials, everything we've done asymptotes at around 15 to 20, and beyond that you don't get a whole lot more benefit. So but the proof's in the pudding. I don't know.
So you went in and you did these forward predictions, and you said that the inverse ones they're indefinite.
Yeah.
How widely do they vary depending upon the kind of the constraints and the whatever the criteria you make? Do they vary a lot, or do they kind of converge on?
Well, the hottest hotspot, when we've played with this, the hottest hotspot stays hot. And it's really kind of how big the region is. Now, I have to say this too. With the monkey, we only had, we had fewer electrodes relative to the kind of dense caps that you'd have with people. So the denser sampling would give us more resolution to answer that question. And that's one of the research goals going forward at York. Oh, okay.
I'm just curious. You said some people have two folds in the cingulate cortex. Are they different? Are they better, weirder? Do we
Don't know. There's one paper we saw that identifies the presence of a paracingulate sulcus, but some association with schizophrenia. But yeah, I don't know anything that isn't associated with schizophrenia. So no, the short answer is I don't know that there's any, because who's looked? Yeah.
It's a hot area for DBS. Singular.
Yeah.
For mental health, like depression and.
Yeah. I mean, I met some colleagues at Sunnybrook Hospital in Toronto who do DBS for depression, like Helen Mayberg and all. And as we talked about this, it just wasn't something they were aware of either, and it didn't seem to influence their diagnostic or their treatment path. Yeah, it's a puzzler, isn't it? All right. Well, thanks so much for your attention, folks.